have our focus on COVID. So next, I want to bring on Trevor Bedford. Trevor uses powerful computers, complex statistical methods, and softwares to be able to, including a tool he developed called NextStrain, to track viral outbreaks, including COVID-19, influenza, Ebola, Zika. He and his colleagues have worked around the clock since the pandemic began, and have worked around the clock over the past week, looking at the Omicron variant, and he's done tremendous amounts of work throughout this. He was recently named both a Howard Hughes Medical Institute investigator and a MacArthur Fellow, which is known as one of the MacArthur Genius Grants. And of course, we're extremely proud of him at the Fred Hutch and the University of Washington. And these recognize his research and his public health contributions in tracking the spread of viruses, particularly SARS-CoV-2. So I want to move right to, as you could imagine, the Q&A line is loading with questions regarding Omicron. And I know that what reassured me, the resource I went to was your Twitter feed during the holiday that was able to very clearly point out what we know, what we don't know. Now, that was 4 days ago, 5 days ago now. So tell us where we are today with Omicron. What does it mean? What are you thinking about? What do we know and what do we not know right now?
Yeah, thanks, Tom. So this feels a bit, unfortunately, like January 2020 all over again, but now like we all get to freak out at the same time rather than kind of having it be a bit for some people. I think the most important details of what we do know are kind of these two different sides. One is that if you just look at this new variant at Omicron, the number of mutations in the spike protein is kind of wild. So other things that have been circulating, alpha, beta, gamma, delta, et cetera, have maybe 8 mutations, maybe 10 in spike protein. That's largely what's given them their advantageous phenotype. So Omicron comes out of nowhere, perhaps from an immunocompromised individual, and shows up with 30 mutations in spike protein. So kind of as immediately as soon as kind of people were able to see that genome, it looks striking, it looks worrisome. We'll know more in about 2 weeks for kind of how well that evades neutralization and so forth, but it already seems like it will be functionally different. Kevin, on the other side of the genome looking interesting, is that we're seeing evidence of rapid local spread in South Africa, and particularly in Gauteng, this province that is kind of the epicenter. And so if you look at both how rapidly Omicron is displacing Delta in sequenced viruses, and if you just look at case counts over time, you get maybe 3x the current rate of spread of Delta. Delta was 2x compared to what the non-variant viruses. And so these two things together have people very concerned for good reason. Basically, in terms of time frame, I'd expect to understand a lot more in about 2 weeks once we have these functional assays in, and we'll have 2 weeks of surveillance data to see actually how it plays out. And it'll probably take more like a month at least to have good evidence of severity. I know there's kind of a discussion saying that it's way less, it's more mild, but I don't think we can say anything there yet, and it'll take time for that data to come in. It could be more mild, it could be more severe. We just won't know.
I would like to bring back Dr. Rachel Bender Ignacio. Rachel's the medical director of our COVID-19 Clinical Research Center, which conducts trials for therapies to treat COVID-19 early in the course of infection. For example, the Merck drug that actually is a drug that treats the virus was something that Rachel was crucial in developing. And she knows about as much as anybody about the state of COVID-19 therapies, and we're delighted to have Rachel with us today. So I guess the first question we're going to ask Rachel is, Trevor just said that COVID's not going away. He also said that he felt that the past week has reminded him a lot of January and February of 2020. And that makes the work that you do in treatment even more important at this point. So what can you tell us about some of the antivirals that you're developing, and do you think they have the potential to change the course of the pandemic?
Yeah, absolutely. Thank you so much, Tom, and it's great to be here. As Trevor was talking about, the Omicron variant has a lot of mutations in the spike protein, but we really don't think that those mutations will have an effect on the way that the virus produces itself once it's inside of a cell. So as a reminder, viruses are not independent living beings, and they use some of our cellular machinery to replicate themselves, as well as some of their own machinery as well. And anything that we call a direct-acting antiviral, so molnupiravir, which is the Merck drug, the Pfizer protease inhibitor, there are a couple other companies making protease inhibitors as well, and remdesivir, which has FDA approval for treatment of inpatients, and we also studied here in preventing hospitalization and severe outcomes in outpatients here at the COVID Clinical Research Center. None of these act on the spike protein. They work on impeding the virus's ability to manufacture itself. And so, of course, while any virus can mutate and does, that's the rule of viruses, is that they mutate. We don't think that these new developments actually will change the efficacy of any of these treatments that we have in development. And I think one of the most really exciting things is that we see that remdesivir, while it works modestly in patients that are admitted to the hospital, works very, very well early on in disease. And I know that the company manufacturing this is working really hard to make an oral form so that we can get it to people, for example, from a pharmacy the moment that they find out they're infected, just like we hope to do with molnupiravir and the Pfizer drug and other oral treatments.
Thank you, Rachel. So I guess one question for both of you is the recognition of Omicron, I think, has led a lot of people to believe that boosters are going to play a larger role. And I'd like to know from both of your perspectives what role boosters play. Trevor, first from yours in terms of levels of antibody, and then Rachel, if you could address the question I get asked all the time, which is the use of different boosters in people who might have had their initial vaccine with one brand or another. Trevor.
So I mentioned in 2 weeks having more data. The data that I'm interested in here is you have individuals who have had 2 doses. You look at their blood, you look at their sera, and you look at how well that neutralizes virus. And you look at individuals who have 3 doses, and you look at how well that neutralizes virus. You compare how well, and we know that 3 doses neutralizes Delta really, really well. And we'll be able to, in a couple weeks, have the assay data of what is the drop for Omicron. It's 2 doses. I'm imagining it could be a pretty significant drop. We know from 3 doses you get this nice broadening of the immune response, so it might be less of a drop with 3 doses, but we don't know yet. And so we could get lucky, and 3 doses and booster will still protect against infection quite well, or we might need to be trying to very quickly have an Omicron-specific booster and kind of move that through the regulatory process. It's also possible that even if that neutralization drop is decent with 3 doses, that you could have a situation where, just like we kind of saw with waning immunity in 2 doses, where you don't get great protection against infection, but you still have decent protection against severe outcomes. That is also possible, and we'll kind of have to wait and see what scenario of the 3 that is.
Okay, and Rachel?
Yeah, I share Trevor's pause in wanting to jump to conclusions about any of the sort of clinical or public health implications about our vaccines or our treatments with regard to a new variant. I do think it's important for people to get boosted in general, and especially in the face of a variant, but it's, I think, more important to get first doses into arms, and that includes people here in the United States who have not yet gotten vaccinated. Any of the 3 vaccines, the series plus the booster is a fantastic idea. And then focusing again on equity, bringing this back to what my colleague Rachel Sacco was talking about, is equity, both within the United States and its global equity for vaccines. We know that we don't know exactly where this variant originated. It was detected first in Botswana and South Africa, but it is an entire region in which we are not, as a global community, doing very well at vaccinating. And so I think, each one of us can talk with people of influence and make sure that we are focusing on global equity with getting first and second shots into arms as well as third shots. Yeah, as we were discussing, we do not think that any of the current variants and the mutations in those variants will have an impact on direct acting antivirals because of where those mutations are within the virus. There is a possibility that these monoclonal antibody treatments, which are some of which have emergency use authorization and are being given for post-exposure prophylaxis or treatment of early COVID, that some of those may have a drop in efficacy, and those are antibody-based therapies, and some of those target sort of similar places to where we know that there are mutations. And we think that some of these treatments, including some of the ones that we've studied at the Hutch, will actually hold up better, again, based on where the therapy matches up with the mutation. But I agree, I think we need to see data in people. It may be possible to overcome some of the decrease in binding and still have good results with these treatments.
So thank you both